Hi everyone, I hope you're having a nice start to your week. This is going to be a bit of a heavy lecture, so let's just jump into it. So the first half of the lecture, I'm going to introduce you to the structures of the eye and the concepts related to the visual system. There's going to be a lot of information here, so feel free to pause and replay as necessary, and please ask me questions in email or office hours. And the second half of the lecture will be related to things you're going to see on the second practical exam. So let's get into it. So before we get into the nitty-gritty, we need to have a baseline understanding of the structures that create the eye. We break up the structures that we can see from the outside and the inside of the eye. So from the outside, we can see the sclera, the cornea, the pupil, and the iris. So the sclera protects the inner eye and stops foreign objects / materials from entering the inside of the eye. It tends to look white, but there are, on humans at least, there are some differences between human and animal sclera. Ours is white and thin, and most animals, including sheep, will kind of have a tannish or yellowish sclera. It's also much thicker. It tends to be an extension of the dura mater that we see that covers the brain that we just talked about a couple of weeks ago, and light does not pass through the sclera. But something that's interesting to consider is that there are some people that have asserted that humans have a whiter sclera to provide another form of subtle, non-verbal form of communication between people. So it can indirectly tell us or tell other people, rather, what we're focused on or looking at. So this extra level of communication is considered worth the loss and protection, since we're social animals and we rely on our community to help us, to figure out what's going on, help us against threats. I mean, it's not really something that as a society we deal with in the same way that animals do, but a significant portion of our brains are dedicated to social, communication, body language, so it's another level or another way that we can communicate with each other. Anyway, moving on to the cornea. This also protects the eye, but in a different way. So here is the cornea. It will also protect the eye from foreign objects or allowing it to damage the eye. But the cornea, unlike the sclera, does allow light to pass through. And it you can kind of think of it like a contact lens. It will also protect the iris. And this is the first place where light enters the eye. And it makes sense if any of you have taken like a first or second semester introduction to physics course, or maybe in high school physics, how light enters the eye. It has to be focused more and more, because outside of the eye light isn't as focused. So then as we're getting to the back of the retina back here, we need to make sure that light is getting focused so we can have or interpret an image of what we're seeing. And now we have the pupil. So it's that dark black portion of the eye. And this is where light can strike the retina in the back of the eye. And finally, we have the iris, which is the pigmented or colored part of the eye. So if you have blue, green, brown eyes, that's from the iris. This controls the size of the pupil or the amount of light that enters the eye. You can think of the pupil as a window that lets light in, whereas the iris is the curtain that determines how much light is coming through. The iris will move depending on how much light there is in the area. So more light means a smaller pupil, and vice versa. And for those of you who are interested in photography, this is the same principle behind how aperture works in a camera. In fact, cameras are actually based on how our eyes function. So taking a look at some of the internal structures of the eye, so if we were doing live dissections, we would have to cut into the eye in order to see this. So when you watch the dissection video after this one, this is what you would see after cutting into it. So we have the lens, which is a disc-like structure that focuses light from the pupil to the retina. And it has muscles connected to it that will allow it to concave or convex, so kind of become wider in the middle or skinnier in the middle. This is also kind of how glasses work, depending if you're near or far-sighted. I believe convex lenses are for nearsightedness. So if you kind of have like it makes a C shape. Anyway, so when we focus on an item in our foreground, the muscles that are connected to the lens will contract and relax to make the item closer to our vision in the foreground sharper, and then the items in the background will be fuzzier. That's also called depth of field for the photography or film people in the audience. And then we also have the anterior and posterior chambers, and these are filled with different fluids. The anterior chamber is filled with aqueous humor, which is, as the name might suggest, aqua, more water. So it's very liquidy in its viscosity, and it helps keep the eye maintain a moistened state. So everything between the cornea and the lens will be filled with aqueous humor, or that accounts for the anterior chamber. And the posterior chamber is filled with vitreous humor, which allows the eye to maintain its volume and its bulbous shape, like its spherical shape. It's kind of like jello in its viscosity compared to aqueous humor. And the lens to the retina, so back here, is the posterior chamber. And these are all the structures that we find in the back of the eye. So we have the retina, and that contains photoreceptor cells, and it takes in light and transforms it into the neuronal signals that we can understand and further process in our brains. And the fovea is where we have our best visual clarity, and information from the fovea is sent to the periphery of the retina. And there's also a blind spot, which I'll talk a little bit more later. And it's approximately 15 degrees toward the nasal side of the fovea, so the part of our fovea that's closer to our noses. And our brains will use information from the other side of the eye to make up for the information lost from the blind spot. I'll go over this in a little bit more detail in a moment, so don't worry if that sounds confusing. So further, we have the choroid, which is the vascular layer of the eye, meaning it has blood vessels, and these nourish the retina. They play a role in absorbing extraneous light so no other structures are damaged, like the optic nerve. So speaking of the optic nerve, these are bundles of axons that form at the retina. So this will send information from the retina to the brain and further into the occipital lobe, where the primary visual cortex is. So this is the second cranial nerve that we had talked about, and the pathway where some visual information is crossed is the optic chiasm. And we'll talk a little bit more about sending information to the lateral geniculate nucleus and the thalamus. We'll talk about the pathways all in a moment. So this is just a magnified image of all of the internal and external structures. So cornea, pupil, iris, these are all in the sclera, are all things that we can see on the outside of the eye. And then we get to the lens, the choroid, the retina, and the optic nerve. These are all things you would have to see with the by dissection. And then here is a visual representation of the different cells that go into how we interpret light. But we'll talk about that in just a second, which is now. So let's dive a little bit more into the retina. So neurons in the retina are involved in the absorption of light and its transduction into electrical signals. And sensory transduction is the act of changing an environmental stimulus into a sensory event. So that's like changing a sound wave into music or light waves into an image. And there are three retinal layers. We have the photoreceptive layer all the way in the back, or the most posterior layer, bipolar, which is in the middle, and then we have the ganglion layer, which is anterior. So this is going to be a little confusing because light will hit the photoreceptive layer first, which is the most posterior layer. It's in the back. You know, as you can see, the cones and rods are photoreceptive layer. Light pierces through, and they will, and both the cones and rods will pick it up first. So then it goes photoreceptive layers to bipolar cells and then to ganglion cells. I mean, I know that the picture makes this look like it's really thick, but I mean, the retina is a very thin structure in the back of the eye. So it's not shocking that light could penetrate through all of these, but it's not necessarily intuitive to think we go back to front when we're thinking about how light hits the eye, or hits the retina, excuse me. So this is the route that light will travel within the retina. So this is how cells respond to light. So we go photoreceptors, which includes rods and cones. They will absorb light in the back of the eye and then send a message to bipolar cells, which are in front of the photoreceptors. The bipolar cells will then receive input from rods or cones or horizontal cells, which I will talk about in a minute when I have a picture. So these will be designated as bipolar rods or a bipolar cone, and horizontal cells lay between photoreceptors and bipolar cells. They have a modulatory role and letting us see whether we have a light or a dim environment. So, luminance, or it allows us to compare. Like, for example, the presentation is white, but the text is black. It helps us see the contrast between them. So then the bipolar cells will get input, the bipolar cells will give input to the ganglion cells, and the axons will form the optic nerve, and then we have amacrine cells that are between the bipolar and ganglion cells, and we don't really know what they do. We know there's a modulatory role to some degree, or they have some modulatory role. It's kind of one of the things that's exciting about neuroscience, that we're still actively learning about different parts of our own bodies and brains. There's still so much that we don't completely understand just yet. So to kind of bring everything that we just talked about together, the best way to remember light hitting the retina goes photoreceptors. So back here, or no, excuse me, this one's flipped. So we have photoreceptors here. Sorry about my kind of haphazard drawing, but so we go from photoreceptors to bipolar cells here, and then we go to ganglion cells that are the most anterior to the retina. So you have to keep in mind that horizontal and amacrine cells act as modulators or regulators, and they are also, amacrine cells are here between our bipolar and our ganglion cells. And our horizontal cells are between our photoreceptive layer and our bipolar cells. You can't really see them all that well, but it's like right here, other ones right here. They're horizontal compared to the verticality of all the other layers. So this is an important slide. So remember, photoreceptors to bipolar, bipolar to ganglion, ganglion to optic nerve. And now we're going to take a little bit more of a detailed look at each layer. So let's take a closer look at rods and cones. So we have our photoreceptors. These are the first that respond to light. So rods are good for showing low levels of light and movement. They're located in the periphery of our eye, and we have more rods than we have cones. So rods are really good for black and white vision. They can tell us about luminance better than cones can, because cones are involved with color vision. They're located very strongly, a strong concentration of them in the fovea, which is the center of our vision, and they work best in bright light. So the high concentrations of rods in the periphery allow us to see low light well. So if you've ever had a moment where you kind of see something in the back of your eye, if you're maybe sitting in a dark room, getting your work done, or watching a movie, and you think you might see something, and then you turn to focus on it, it's harder to see whether or not it's there, because cones work best in bright light. So it's hard to see what's going on, but it'll be much easier to see in your periphery. And the paper squares example will be tied to the lab report. So now let's take a look at ganglion cells. These cells release neurotransmitters, but they do not absorb light like photoreceptors. And we have several different types of ganglion cells. We have M and P. And there's also a less talked about K cell. I'm not going to test you on it, so don't worry. But when I was learning about this, I also learned about K cell, so pass on the information. Even though they're ones we don't know as much about. And I'm going to make the font a little bit larger because it's smaller than what I expected. Sorry about that. I noticed that the smaller bullet points were a little hard to read. So first we're going to talk about P ganglion cells, which are parvocellular ganglion cells, and these are responsible for fine details and color information. Mainly used to determine small details of a stationary object. They have smaller cell bodies and small receptive fields, so more specific in what they'll pick up. And they're mainly located around the fovea, and they have high spatial resolution. So they're able to pick up fine details, which would make sense. They would have better spatial resolution because they're more selective, having a smaller receptive field. Next, we have the magnocellular ganglion cells, and these are responsible for movement and broad outlines. So they have larger cell bodies, large receptive fields, and they're located more in the periphery, and they're not color sensitive and low spatial resolution, which makes sense. If you have a large receptive field, then you're going to pick up everything that you see, so you're not going to be specific on what's going on. Specificity comes from a smaller receptive field, and will in smaller receptive fields increase spatial resolution. So M and P cells work together simultaneously. Nothing works by itself in the brain ever. In addition, we also have the coniacellular or K cells, and they process other aspects of color. We don't necessarily know a whole ton about them just yet. There are some indications that they work alongside like color contrast. So like P cells do fine details in color. K cells might work with the contrast between different colors. I just thought that was an interesting further knowledge, but I won't test you on K cells, just M and P cells. And I remember M and P cells, so P for picture, fine details, color information, and M cells for movement because they take care of movement. So now we're going to talk about the optic nerve. So the optic nerve takes space where rods and cones would be, and which results in the blind spot that I talked about earlier. We need the rods and cones, a photoreceptive layer for the eye, for us to process light in the eye and have it be turned into a neural signal. So this is where our ganglion cell axons will bind together and form the optic nerve. And the blind spot is where ganglion cell axons will leave the eye. I will put a link to the blind spot test. I will show it to you really quick. So I'm having some problems getting my screen capturing software to pick up the entire site. However, these are a bunch of tests to show practically where your blind spot is. So I'm going to leave a link to it in the description. So do not worry. I do encourage you to check it out, though. It is really cool. So moving on, we're going to talk about the reticulogeniculate striate pathway. So there are three main stops from our retina to the visual cortex, or the striate cortex. So we're going to go from retina to thalamus, which specifically is the lateral geniculate nucleus, or the LGN. Please do remember this. And then to the primary visual cortex, or V1. So the primary visual pathway that will convey information is essential for visual perception. So in our picture here, let me make this a little bit bigger. We're going to go, light will come into the retina, into the eye, and travel down the optic nerve. And as you can see, we have the green and the red. So red represents the nasal visual field in the temporal retina. Don't really worry about that, but needless to say, at the optic chiasm, red will cross over to the right side of the brain, and then green on the right side will then cross over to the left side. So this is the area some of this the visual information will cross over. And then that information will travel to the thalamus, to the LGN, and then over to the primary visual cortex. That's the kind of the first step pathway. There are more steps along the way, but these are the three main stops that we need to worry about. Should make this a little bit smaller. So we have the optic tract, which is in the central nervous system, which will project to the LGN of the thalamus, and the LGN will project directly to the primary visual cortex, or V1. And I wanted to include this picture simply because it describes how our vision crosses. Actually, make this a little bit larger. So you can see some of our visual field on the right and left sides cross over, so that we're getting information from both sides to get a more complete picture, because it covers the spots where our blind spot is. So we're taking information from our left eye to compensate for what we're losing from our right blind spot, and vice versa. And I like this picture too, because if you look at the red lines or the boxes, it shows you, like, if I cut at area 1, then you would have blindness in the right eye but vision in the left. If you cut at the optic chiasm, then you're only going to have vision in the temporal retina, so in your periphery. And then same thing if you're going further down the optic nerve, or the optic tract, since we're in the central nervous system at this point. And these are all just really cool ways to think of, if you lesion different parts of the brain or the nerve, how will that affect your vision? If you have any questions about this, please let me know. About any of this, obviously. Anyway, so let's talk about our second to third stops in our retinogeniculate striate pathway. So going from the LGN to the primary visual cortex. I'll probably call it primary visual cortex, or V1. Striate cortex is another completely valid name that you'll see in papers. V1's another one. And then area 17 is the Brodmann's area associated with V1. And it is and V1 is part of the occipital cortex, is part of the first stage of visual processing, and devoted to special features of vision, pattern recognition, orientation, movement, spatial frequency, and color vision. Some of it, not all of it. So more visual processing will occur in V2, and then, which is separation of multiple objects, and then we have to V3 and V4. So V3 is involved in the orientation or the motion of an object, and V4 is very important for color constancy. So being able to tell an object will retain its color under different lighting conditions, very, very important. We'll talk a little bit more about these in a moment. And so V4, if it's reliant on information about color and color constancy, V4 relies on information from our cones rather than our rods, which are responsible for black and white vision. So signals from V1, V2, V3 will go to areas V5/MT, and then later to MST, which is the medial superior temporal area. So MT, or the medial temporal area, is part of the dorsal stream, which I think we'll talk about in a little bit, which is devoted to the analysis of movement, so acceleration, deceleration. And then MST is adjacent to MT, involved in processing the expansion, contraction, or rotation of visual scenes. So you can think MT, moving on the X or Y axis, I guess, if you're moving up and down, and then left and right for X axis. And then MST is kind of on the Z, where it's like roll and pitch and yaw, like how that's moving. Anyway, let's look at color constancy. We have a singular apple here. So as I mentioned before, color constancy is our ability to perceive colors as being constant over varying light. So this apple would still be red if I looked at it in the grocery store versus in a shaded, like under a shady tree. So color constancy is actually one of the ways that marketers can use to change our perception on the items that we buy. So part of the reason why grocery stores and other stores have really bright lights, but specifically grocery stores, it's not just to let us see clearly and easily read the ingredients on your products. It's to make produce look as fresh as possible, even though it might not be as fresh as it is. So it's why it's good to really inspect what you're looking at before you buy it. It's why you can pick up some apples or tomatoes. You're like, oh, this looks really good. Can't wait to have it later. And then you're at home in different lighting conditions. And you're like, wait, this is a nasty piece of produce. Maybe not that extreme, but color constancy is certainly one of the ways that marketers can try and manipulate how we view certain objects. So here we are to the dorsal and ventral streams. So this information is sent to the extrastriate areas in the parietal, temporal, and occipital lobes. So this is related to the stuff we were just talking about from the LGN to V1. So these streams will diverge. So you go to V1, and then after V2, our streams will break into two main streams. So we have the ventral stream, which is V1, V2, V3 to V4, and then it goes to the inferior temporal cortex, which the what pathway, as the name may suggest, is responsible for identifying and recognizing objects or shapes, and they provide visual information about size, shape, color, and texture. So what is the object that we're looking at? The dorsal stream, on the other hand, is the where pathway. So it goes V1, V2, V3, but instead of going to V4, it's going to the medial temporal lobe, or V5, to the posterior parietal cortex. So I'm going to highlight both of these since that is the main change here. And the where pathway processes movement and spatial location and provides information about what guides navigation and skilled movement. So where is something in your visual field? So dorsal, where, ventral, what? In this picture on the left here has a visual representation of, we start at V1 in light blue to V2, then we go to V3 in green. And then if we're going with the ventral pathway, we're going to V4 and then out to the inferior temporal cortex by the ears. But if we're going to the dorsal pathway, then we're going to MT and then further to the posterior parietal cortex in the parietal lobe. So we've talked so much about pathways, color constancy, the structures of the eye. What are some of the ways that this can all go wrong? So blind sight and visual agnosia are two really common disorders. We'll cover a couple more. There are tons of ways for sight to go bonkers. I mean, as you can see, these are just physical lesions that can occur and change things. In the LGN, that's not even mentioning like V1 or like problems in V4 or in the dorsal or ventral pathways. So anyway, let's talk about blind sight and visual agnosia. So with blind sight, the patient is no longer aware of what an object is, but they can still grab it. So that would be like, I have my water bottle in front of me. So I couldn't tell you that the water bottle was there, but I could pick it up. So there's no conscious awareness that the item is in front of me, but I can still like physically see it. So you can't name the object because you can't see what it looks like. And this can be a result of damage to the occipital lobe, so V1. So our like the eye's work, all of this is functioning, except when we get to V1, which is processing our like the basic parts of what is actually coming from the retina. So scientists believe that there are some aspects of visual processing from the superior colliculus, which, as you may know or should know, is responsible for simple aspects of visual processing. And visual agnosia, which is a higher processing disorder, sight remains intact. It's the inability to identify objects that are visually perceived even though the object can be drawn or matched. So I couldn't tell you what the item was verbally unless there's another input. And this is caused by temporal lobe damage. So this is what pathway damage. So if I were to ask a question like, what would be an example of what pathway damage, then you could say visual agnosia, because you can't identify what the object is without using another sense. So I'd like touch or hearing or something like that. So to talk a little bit more about some other disorders, we have prosopagnosia, which is the inability to see people by their face. This is face blindness. So you can recognize that you're looking at a face, but you can't put a name to it. You don't recognize the person. And this normally occurs because of damage to the fusiform gyrus and the temporal lobe, which is our face recognizing, well, part of the face recognizing areas of the brain. And if any of you are familiar with the famous neuroscientist Oliver Sacks, he wrote a book called The Man Who Mistook His Wife for a Hat. It is a famous case of prosopagnosia where a man would literally think that his wife was a hat if she was in certain parts of his visual field. And if she would stand to like in the left periphery, she would say, "Oh, that's the new hat that I bought," or something like that. Like, I knew someone actually with this disorder, and they would get to know you over time through episodic memory. But if you would change something significant, like wear a different hat, like change your hair significantly, this person would not be able to recognize me unless I maintained a very similar look throughout the time that we knew each other. So it's a very interesting disorder. And finally, there's motion blindness. So you can still see, but you can't detect motion, and this can be a really big problem if you're crossing the street or doing things like that, because you won't be able to notice that a car is moving toward you. So like the car would kind of almost teleport. So it would go from like 5 feet away, 2 feet away, 1 foot away, but it wouldn't have a smooth transition. And this is normally from damage to V5 in the occipital lobe. So damage to the Ware pathway. So right here. So I imagine that would cause damage to the pathway from V5 to the posterior parietal as well. So these are just some of the common sight disorders. There are tons of others. There's a lot that can go wonky when the visual system is concerned. So now we can take a little breath, like, whew. Talked a lot about the eye, a lot of pathways, lots and lots of information there. And now we're going to look at some anatomy stuff that's going to be on practical too. You'll not need to know this for the quiz this week, but you should have an understanding of the functions. So I'm not going to give you any eye pictures to look at for quiz 2, but do understand what the structures do, please. So let's take a look. So these are all, this is an outside structure this is the nictitating membrane. This is not found in humans. It's a third eyelid. So like if you see with cats or with dogs too, but in this case, this is a cow eye, I believe, or a sheep eye. It will protect the eye kind of while you're sleeping. So if you've ever seen a dog or a cat kind of sleep with their eyes open, you're looking at their nictitating membrane. And this is controlled by the abducens nerve. And then we have the sclera, which humans do have. Ours is whiter than animals. So you can see, I mean, aside from this is a preserved eye, this would be whiter in humans anyway if we were dissecting a human eyeball for whatever reason. So this will be the white protective covering, not found in the cornea, and sclera does not allow light to pass through, and it's an extension of the dura mater. We have the cornea and conjunctiva. So the cornea is similar in its protective tendencies like the sclera, but it does allow light to pass through. This is the first spot where light passes through. And the conjunctiva is inside of the eyelid and provides lubrication, and it's located at the junction of the sclera and the cornea. So where the two meet, that's where you see the conjunctiva. And if you have an infection here, you have conjunctivitis. And moving to the vitreous humor, these are going to be inside of the eyeball, so you would have to, as you can see in the image, cut it in half. So vitreous humor is in the anterior or posterior chamber. Just give a second. So I hope that you said posterior chamber. Anterior chamber has aqueous humor. And vitreous humor is a gel-like substance, and it provides shape and support, so it allows the eyeball to keep its round spherical shape. Its bulbous shape, if you would prefer. And then we have the lens, and this will refract light to help focus images near or far. And when it's stretched, it helps us to see light far away. So this image in the bottom left just shows how the lens can be accommodated, so the muscles will pull it taut, and then the near or images in our foreground will be blurry, far images will be in focus, and the vice versa. So when you compress or concave it, or convex it, I believe, but when you're not pulling it taut so that it's long and thin, that helps images to be in your foreground to be in the focus, and the far in the background will be out of focus. And this is how light refracts from lenses in the brain. If you've been, as I said, in a like introductory physics course, or you might have done something like this in lab with lenses. It's the same process for our own eyes. And the lens, especially after preservation, feels really hard, almost like a marble, and is surrounded by vitreous humor. So moving on to the retina, that is all the way in the back of the eye, and that's neural tissue containing our photoreceptive cells. It's located on the inner surface of the posterior portion of the eye, and it might be a little hard to see where the pin is. I'll try to circle it. It's kind of like a cream whitish color. And it's only connected to the eye via the optic nerve. And then we have the choroid, which is a black substance on the back of the eyeball, and it's located behind the retina and absorbs stray light so that we don't see images or information that we don't need, that gets refracted from the lens and from outside. And further, that the choroid provides nutrients and oxygen to the retina through blood vessels. And we have the tapetum lucidum. This is not found in the human eye. It's something you see in animals. So whenever you're trying to take a picture of your kitty or your puppy, or sheep or cows, I mean, all animals are awesome. When they have that iridescent quality, like those glowing eyes, you're getting a picture of the tapetum lucidum. It's because that's the iridescent membrane. It's a reflective structure to allow for better night vision. It's found in sheep, alligators, cats, as I said before. we gave this up basically for better room for other senses. But it's important for animals, especially cats. Cats have a very strong predator tendency. They love to go hunting and like bring back mice. And if any of you are cat owners, I think you'll know exactly what I'm talking about. They will love to show you what they're hunting. Having this iridescent membrane makes it really, really easy to see whatever stray light they can at night. Because without it, you don't have any reflection of just the teeny bit of light that you can see at night. And finally, we have the iris. And this is the pigmented part of the eye, so blue, green, brown eyes. Has a sphincter and a dilator muscles that will control the size of the pupil. And look for everything on the picture, on the eye that's pictured to the right, plus the nictitating membrane. So here, on our preserved eye is the iris. That hole is the pupil. And it doesn't look black because we're not looking toward the back of the eye. We're just looking straight through it. So you can see the background through the pupil. All right. So this is all of it for the visual system. We covered an absolute ton in this lecture. So please reach out in office hours or email if you have any questions about any of the pathways or the structures or really anything. A visual system is complex on a good day, so there's no worries if you're confused about something that we talked about here. And for today's, or this week's rather, attendance word, we're going to do blind sight, I think. So from the sight disorder slide, blind sight. So if you just spell it just like this, that is our attendance word for this week. So this week we have a quiz and a lab report due. Please let me know if you have any questions or comments or anything like that. Take care, everyone. I hope you have an amazing week, and I will see you next time. Bye.